right? So if you have any questions, uh, feel free to stop me and ask questions. Yeah. So the first part is going to cover a very basic structure of the lateral flow, or usually known as test strip, okay, rapid test kit. So there are other names, but the standard name is lateral flow. So what is actually lateral flow? So the full name is immunochromatographic assay because it looks like a paper chromatography. So if it works like paper chromatography, the separations. So it's a rapid test, yes, because of the time taken for us to have the answer yes or no, because it's not qualitative. It can be quantitative, depends on how you design the rapid test kit. But at this moment, I can only share with you the qualitative yes or no rapid test kit or lateral flow. And the type of sample, we will discuss this in the second part of my talk. So the first hour will be on the structure of the lateral flow body. And the sensitivity of the lateral flow depends on how you design, how good you design, and also how good your biomarker is, and also related to how good your nanoparticle is. Okay, most of the time we use gold nanoparticle. Yes, it's common, but there are other nanoparticles that can be used. For example, quantum dots, carbon dots, fluorescent, which is more sensitive compared to gold nanoparticle. But at this moment, for the purpose of our training, we focus on gold nanoparticle. Okay, so the first is the structure. Okay, so as I said, I'm not sure how deep your um, basic in lateral flow, but for those who has advanced knowledge on this, please bear with me. I have to cover everybody. So this is the basic structure of the lateral flow, or the test strip, known as test strip. First, we have the sample pad where we put the sample. So everybody, I think, has used this during COVID, where we put the saliva here. Okay, so this is where we put our saliva sample here. And then move on to conjugate pad, where we have the biomarker, the capture antibody, and then uh, the whole thing will move, okay, towards the test line. So if there is interaction, meaning that there is the targeted virus, the targeted protein, or the targeted bacteria in the sample, then there will be interaction of the targeted bacteria, targeted virus with the antibody here. Then the test line will appear, and also the conjugate pad, the capture antibody in on the conjugate pad also will also interact with the control line. Antibodies that we put in the control line usually is the general one, is the IgG, IgM. So the wicking pad is just where all the rest of the samples are uh, actually pooled here. So the backing pad is just a support. Yeah, it does not play any role in the process of the during the movement of the sample. So in detail, very important. Okay, as I mentioned, sensitivity wise of the rapid test kit for lateral flow depends on the design of the lateral flow. So if you ask about design, there are two types of design, meaning that, sorry, there are two factors, biggest factors. One is how well you manage your conjugate pad. Okay. Also, the sample pad, okay, how we prepare the sample pad, and also uh, the length. The other factor is the architecture of the lateral flow, meaning that um, the length, okay, from here until here, uh, you can design the length from here to here, and how long you want the sample to travel before it reach the test line, okay, the control line, because we are working with different type of sample. You might be working with serum, you might be working with urine, or you might be working with whole blood. For us, we have experience working with sputum sample, which is very viscous. So sometimes it's difficult for the sample to move from here to conjugate pad and move to test line and control line. So the design or the architecture of the lateral flow is very, very important for any researcher to characterize first, to optimize which length suits your sensitivity. We have difficulty at the beginning to get a very sensitive reading. So after sometimes, well, due to experience, finally to get the nice reading, because it depends on how sensitive your detection is, meaning that how low that you need to detect the biomarker, the protein, the virus, the bacteria, how low you need to detect. So you have to characterize the architecture of the lateral flow as well. So it's not just about how much biomarker or how much, for example, capture antibody here, how much uh, the capture antibody here. It's not about how much, it's about also the architecture of the lateral flow. Okay, so tomorrow you will be learning the hands-on. There are many types of what we call as uh, all these pad. They can be assembled, okay? And this padding is based on nitrocellulose membrane. And there are other types of membrane that can be used. And even this nitrocellulose membrane also have different pore size. Okay. So if you want to study, okay, the for your own application, I suggest that you buy a different set of nitrocellulose membrane, okay, because they have different pore size, they have different characteristic, and do some characterization before finalizing on, okay, this nitrocellulose membrane type is suitable for you, because that will uh, your sample, okay, will have some effect on the suitability of your membrane. Okay, so it doesn't like one formula fit everything. No, we have to characterize the architecture, the length, and also the amount of antibody that you need to put here. Okay, so that is something that you need to remember when ordering the basic like membrane or all these paddings. You have to think about exploring different types and different sizes of pore sizes. Okay. Right. I think this is something that the basic what is happening. How do you get the test line? How do you get the control line? Okay. So this is where the sample comes in, and you have the conjugate pad. And when it moves, if it is positive, then the capture antibody will also capture the antigen of interest, and which also conjugated with the uh, antibody and the gold nanoparticle. So you have a test line occur, okay? And the same conjugate also will interact with the commonly used IgG, IgM antibody, okay? And that's how you get the control line. So if there is no interaction occur with the capture antibody in the test line, so it should not be, it shouldn't be any color or any signal generated in the test line, whereas for the control line has to be there because this indicate that the lateral flow is working. It's not uh, it's not damaged or broken, but the control line is to confirm that the lateral flow is working, but it is not, it is a negative result. Okay, so basically, this is what is happening on the lateral flow or test strip. Okay, so this is another picture showing how it happened. Okay, uh, negative and but this one is positive. So the better picture, all right? So coming back to the architecture of the lateral flow, okay? So the padding, the solid support, okay, there are certain different types. For example, we have commonly used nitrocellulose membrane, okay? And as I mentioned, they have a different porosity. So you have to characterize, okay, to suit your characteristic of your sample. Some sample might be uh, very viscous, some sample might be very thin, or some sample requires filtration. For example, if you are working with whole blood, okay, because the red blood cell will definitely interfere with the uh, color in the test line as well as in the control line. So you need some additional features, which is the filtration of the red blood cell. So if you're not working with something colored, then it will not interfere. But if you have colored compound, then you have to think about the filtration here, right? So there's another one, nylon, which we are not familiar with. And if you ask tomorrow, we are more on nitrocellulose membrane. Yeah? So advantages of nitrocellulose membrane, yes, it's low cost. Okay, it has a good capillary, but as I mentioned to you, you have to check in terms of porosity, high protein binding, ease of handling. Yeah, based on our experience, yes, this is something that uh, you can observe from that, from the usage of nitrocellulose membrane. Okay, conjugate pad. Okay, so this is where the conjugate, so sample pad and conjugate pad. So this is where the conjugate pad, very important. Okay, this is where you put the uh, conjugate of gold nanoparticle or any nanoparticle that you're working with, which you conjugate with the antibody, or it might be antigen. But tomorrow we will focus on antibodies because we use sandwich type uh, detection. So this is where you put the gold conjugated with antibody. And this is where you need to characterize in terms of concentration. Okay, how much you need to put the conjugated gold antibody on top of this conjugate pad. Definitely, working with antibody is very expensive, especially if you're working with monoclonal. Polyclonal probably cheaper, but monoclonal is expensive, and you have to really optimize how much of your conjugated antibody here, because one thing, of course, you want the best test line, the most readable test line that is easy to be look at with naked eye. But at the same time, we need to save on cost. Okay. Based on our experience, the cost of lateral flow actually the biggest goes to the antibody. Yeah. The gold nanoparticle, because we prepare uh, uh, in our own lab, so it's not that expensive. We can get a lot based on one preparation, but the problem is the antibody, because if we use a very low concentration, then we might not be getting the test line at all. But if we have it also cannot use so much. I mean, if it is highly concentrated, then it will also inhibit the reading. So the optimum, okay, based on our experience is, but you may ask Dr. Hafiz and Dr. Ain tomorrow. Uh, Theoretically, it should be not more than 1 milligram. Okay, I do have the, yeah, it's not more than 1 milligram per liter. Okay, so because if you are using sandwich base, okay, you need to have a lot of conjugated antibody here. So based on the tutorial that I have provided to you, there's one paper under Nature Protocols. Okay, you can read in the tutorial. There are some guide of how much concentration of conjugated antibody that should be uh, immobilized here. Okay, right. So how to conjugate the antibody with the goal, you will learn tomorrow. Okay, so it's a simple chemistry. No need to think about complex complex chemistry to bind the antibody with the gold nanoparticle. There's you don't need to worry that. It's a simple chemistry. We thought at the beginning when we worked with this lateral flow, we thought of using a complicated chemistry, but when we do that, it does not perform well. So finally, we just use a simple chemistry. So it works. So no need to worry about the complicated chemistry. Now, the next one is the preparation of the gold particles, gold nanoparticles. Okay, so this one, based on our experience, okay, 40 nanometer is the best size for you to observe a very good test line. Okay, so the preparation of gold nanoparticles, there's a lot in has been published a lot, but you can we can share with you tomorrow the formulation that has been used, and based on our observation, 40 nanometer is the best for you to get the best test line reading. Yeah, and very important, if you prepare gold nanoparticle, please don't stock up a lot because it's not that stable. Yeah, it's best for you to prepare in small batches because it needs to be fresh. And the best is every time you want to prepare the lateral flow, you need to check on the stability of the gold nanoparticle solution. Okay, you every time you have to check whether they are still stable or they are already aggregated. So that's why you need to prepare in small stock so that you will not waste the gold solution. Okay, so this one is very important because this is the key that give the color on the test line and the uh, control line. So if the gold nanoparticle is not to the optimum size, then you will not be getting a good reading. Okay, so that's why every time you want to prepare a new batches of lateral flow, then you have to check on the stock of your gold nanoparticle, whether it is stable or you need to prepare a new one. So how do you do that? You need to check using UV, visible spectrophotometry, which is tomorrow you will learn on how to check, okay? Right, the sample pad, okay? So back to the sample pad, just now is conjugate, and we talk about gold nanoparticle. The sample pad, where you put the sample. To accept any kind of sample, whole blood, feces, tissue, serum, sputum, any sample that you can think about. Yeah, sometimes it can also use for drug detection. And the sample, also we have cellulose, sample pad, cellulose, glass fiber, and rayon, I don't know, but we use cellulose. And this one also must be treated. Yeah, the sample pad also must be treated with proper buffer, surfactant, or if needed, blocking reagents. Okay, so this one depends on your application and your sample. Okay, so if the assay buffer requires you to prepare certain pH, then you have to prepare the whole thing first before the analysis can be done. Okay, so this part, which is on the buffer, surfactant, or blocking reagents, has to be characterized. Okay, if not, then you will not get the reading. Okay, so it has to be. Okay. Okay. Right. Okay. So the wicking pad, I think there is no issue on this because it's just towards the end of your, okay, towards the end of the where everything is pulled. Okay, but it should not release the fluid back into the assay because it should be the end. Yeah? It should design to pull all of the fluid added to the strip and to hold it. Should not release the fluid back into the assay. So this is the the wicking pad. So cellulose has a high capillary uh, system that should move everything towards here. So we know about capillary effect, but it also depends on the pore size as well as the viscosity of your sample. Okay, so very important, if your sample is too viscous, that's why you need to have a proper buffer so that it can move towards the end. But the issue is if you put a lot of buffer, too much, then it might dilute your sample. And due to the sometimes because test strip is not like it's not too sensitive, it's not sensitive enough like benchtop machines. So if you do too much dilution, then you might not be getting reading here because they might be moving too fast, one thing, that the capture antibody don't have the time to capture the antigen, or it might have not enough antigen in the sample because you have diluted too much. Okay, so be careful. You have to um, optimize the amount of buffer so that it can move properly towards the end. But at the same time, you maintain a suitable amount of antigen inside the sample to be detected by the capture antibody. So those are things that you have to be careful and do a lot of optimization until you get the formulation. All right. And the viscosity of the samples, especially for those who are working with uh, sample that is very viscous, uh, yeah, sometimes the problem is that it cannot move towards the end. It probably stops here or stop in the middle. So that's something that also has to be uh, counter. How do you balance the dilution and make sure that the sample is thin enough for it to move along the lateral flow? So this is based on our experience. And the backing pad is actually just a support where you put all the padding, the conjugate pad, the sample pad, test line pad on top of the support. Okay. All right. So labels for detection. Okay, this one I think the process that you will do is conjugating the nanoparticles, whether it is gold or whether you're using something else like magnetic nanoparticles, or you're using um, quantum dots, or any fluorescent materials nanoparticles with the antibodies. Yeah. So the process, as I mentioned, depends also on the chemistry. So right now we are using a very simple chemistry where we conjugated the gold nanoparticle with the capture antibody. So there is no complicated chemistry at this moment. So this is an example. Just so this is just an example of what contained in the rapid test kit based on lateral flow. So this one I share with you, the one that is being has been commercialized by this company. Okay. So the common thing that we also find in our COVID test kit, okay, you have the diluent tubes, which is representing the buffer, and the lateral flow test kit. You have swabs for the samples, droppers to drop the sample, and of course instruction manual. So if you plan to commercialize uh, the your rapid test kit, then you need to have all this. Yeah. So this is also based on our experience, and this also based on the existing test kit that I have shown just now. Okay, the sampling of these samples, okay, is a bit tricky. Okay, so this also requires you to think about the best protocols for you to get the most antigen from the samples. Okay, antigen means the one that you want to measure, the virus, the bacteria, the protein. Okay, so you have to think about the protocols that enable you to have the optimum amount being collected. Okay, sometimes if you just like use what we call as a dropper, you for example we are working with sputum. If we just use dropper to suck the sputum from the vials and directly put on the sample pad before we put some buffers or any other chemicals, it seems that we don't have enough antigen. Okay, it seems that we don't have enough antigen because we can imagine that in the samples, we are not sure which part is highly concentrated with antigen. We have the sputum samples or the saliva samples, but we do not know which part in the vials that is highly concentrated with the target antigen that we wish to have. So after sometimes we come up with a new protocols to to get the most antigen from the sputum samples. So you have to do that with your own samples. Let's say you're working with blood samples, serum samples. You have to think of a protocol so that you obtain the most concentrated antigen, because working with test rate, they are not sensitive. So you have to optimize your sampling procedure. Okay. So based on this, because they are working with brain sample, okay, they are for rabies. So what they did is using the disposable swab, they swab the brain sample first, okay, and this and that I think add a small piece of the brain tissue into the assay diluent tube. Yeah? So once the first step is they swab and probably they get some brain tissue and they put it in the vials, which contain some diluent, okay, or probably buffers, okay, and because it's a solid, they have to homogenize the tissue using the swab until the sample has been dissolved. This is just manual homogenizing by using the swab, okay, right? So the next one is using the dropper to get the swab, to get the sample from the vials with the diluent and then put on top of the sample pad. Okay, so 3 to 4 drops of sample suspension shall be added. So I think because they have used proper diluent, they don't need to use any other buffer to make sure that the sample move to on the lateral flow. So test results, yes, this is quite average, which is around 20 minutes. You can also reach to 30 minutes. That depends on your samples. If your sample is very viscous, then it will take some time. Yeah? So a bit on fundamental, uh, because as I mentioned, I do not know your background. Uh, uh, Sometimes we can use competitive assay or non-competitive assay. So tomorrow you will be exposed to non-competitive assay. We have not done any competitive assay. But to my knowledge, from my reading, competitive assay is supposed to be uh, much more sensitive, yeah, and use less antibody compared to non-competitive assay. So it's your choice. You can either choose competitive or non-competitive assay. So this is competitive assay. So competitive assay, if you look at the structure of the lateral flow, the difference, the biggest difference is on the test line, okay, we don't put capture antibody. Instead, uh, they put the target analyte, which is the antigen. Okay, they put the target analyte, and then what happened is, okay, please look at the structure. If there is no target in the sample, okay, this is the conjugated antibody, and then if there is no target, you can see that all the conjugated antibody will interact with the target antigen. So you will see a very strong line, a very concentrated line. Okay, so that is for negative test. This is competitive assay. Okay, let's say if there is target antigen in the sample. Okay, so the conjugated antibody will be competing. Some of the conjugated antibody will interact with the antigen that is in the sample, and also some will interact with the antigen on the test line. So since there is a competition, the line here will be very faint compared to the negative test. So this is the concept of competitive uh, test for lateral flow. So I don't have any experience, neither I don't know whether Dr. Hafiz has, but tomorrow we will introduce to you the non-competitive. Okay, so but to my knowledge from the reading, this is more sensitive and use less antibody compared to the non-competitive. All right, so competitive assays are generally used for small target molecules. Okay, so there is a good thing about competitive assay is if you have a target analyte of a very small size and they don't have many binding site, for example, drugs, toxins, okay, it doesn't work like virus, bacteria, they are bulky. So competitive assay is very useful if you're working with this type of sample, drugs, toxins, or any other small target molecules, protein probably, small protein. So you need to use competitive assay. And the detection, the concentration of the detection means that the antibody use is generally between 1 and 0.1 milligram per mil. Okay, I mentioned just now milligram per liter, it's milligram per mil. Okay, just a minute. So you see that the advantage of using competitive assay is for small target molecules and the usage of antibody 1 to 0.1 milligram per mil. Right, so it does use less than the non-competitive assay. So this is the non-competitive competitive assay, and usually is in sandwich mode. Yeah. So this is the competitive assay. So the non-competitive assay usually it is based on sandwich mode, where you have the capture antibody first and then expose with the antigen. And the next one is conjugated antibody with the gold nanoparticles coming in to complete the whole sandwich, and you get the test line. Okay, so by using this non-competitive, you can use for big size analytes or analytes that has many binding sites with the antibody. For example, big proteins, bulky protein, bacteria, viruses, cells. Okay, so that is suitable for a non-competitive uh, scheme, yeah, non-competitive, which is sandwich type. Yeah? And sometimes you wonder whether you should use monoclonal or polyclonal. Based on my reading, of course, monoclonal is much more selective, but of course, it is expensive to produce. Uh, it is suggested that better if polyclonal because polyclonal they have many more binding sites for the antibody. Okay, so I don't know, that one is actually has to be characterized by you. Okay, whether you want to use polyclonal or monoclonal. Okay, we have experience using monoclonal as well as polyclonal. Based on our experience, based on TB, tuberculosis that we are doing right now, the polyclonal works well. And the monoclonal also works well. So, we opt for cheaper option, which is polyclonal. But you see that the concentration of the antibody that is required for non-competitive assay is more than 1 milligram per mil to maximize the chances for the sandwich formation. So this is based on my reading that you can see that the difference just now, if it is if it is competitive, you can use between 1 and 0.1 milligram per mil, whereas for non-competitive, it is more than 1 milligram per mil, which is a lot. That's probably the the uh, the negative side of using non-competitive. Yeah, but it works well with big proteins. Yeah, right. I think I should be going a bit faster. Okay. For the second part later, I need you to at least have with you the nature protocol tutorial because I'll be referring to table 1, table 2. They are all from the nature protocols tutorials that Dr. Intan has provided for you in the email. Okay, so I think this one, okay, right? Preparation, synthesis, okay. So I think this one is the detail, which is something that you will learn again tomorrow. So I'll go very fast. The preparation of sample pad, okay. You see that it's using pet buffer, drying, okay. And I have to tell you that the lateral flow process is very sensitive to moisture, meaning that once you have done the conjugation pad, sample pad preparation, everything has to be in dry condition after that. Yeah, drying at room temperature under vacuum. So you may ask Dr. Hafiz or Dr. Ain tomorrow, um, how sensitive it is towards the moisture of the condition of the room. Okay, because if it is damaged due to moisture effect, then the whole thing will not be able to use. The lateral flow will be damaged if it is exposed to moisture. So that's why preparation room has to be in dry condition. It's not clean room, but it's dry condition. Okay, right? Okay, you see there is drying process. Okay, so this will be tomorrow. You will see the hands-on, and you can involve in the hands-on making of the lateral flow tomorrow. Okay, so this is the preparation of basic gold nanoparticle, okay, using gold salt. In this case, we're using chloroauric salt. And then we have the reducing agent, sodium citrate, right? And the whole process. The preparation of the antibody and the conjugation of the antibody with the gold nanoparticle. Okay, right. So while I am sharing the second slide, I hope that you can download or prepare yourself the tutorial nature protocols. Okay. Any questions? Stop share this one. I'll be sharing the next one. Okay, so this is part 2. Okay. So this table 1, page 4, table 2, page 5, they are all from the Nature Protocol paper titled Tutorial, Design and Fabrication of Nanoparticles Based Lateral Flow Amino Acids. So you can refer all this uh, with the document, with the paper that I have provided. Yeah. So there are very details nature protocols that you can refer to. If you read that, I think combined with the hands-on, it should be, you can start your own research, okay? Right? So this one, I think need something that we have talked about, right? That's 9. Okay, so according to the tutorial that I have provided in this page 4, table 1, okay, before you decide on the structure or the architecture of the lateral flow, you have to get to know your own samples. Okay, so there are many different types of sample: whole blood, serum, sweat, urine, you can name it. Okay, and you need to think about whether you need to have pre-treatment before you can put on the sample pad. okay, or you need to have some filtration system to be employed together with the lateral flow, and suitable buffer solution as well, according to the pH that suits the analysis. And sorry, and all based on the discussion just now, I'm talking about antibodies, but as you can see now, we can also use DNA, we can also use aptamer and any other probably different biomarker for the purpose of to construct lateral flow. So this you can refer to table 2, page 5 in the tutorial paper. Okay, there are many selection of bioreceptors. It's not just antibody. Okay, uh, as long as the bioreceptors can maintain its original structure after wet and drying cycle. Okay, so because we need to employ on the uh, nanocellulose padding, sorry, the cellulose padding. So let's say if we put the aptamer there, the DNA there, you have to make sure that they maintain the original structure. If the structure is damaged, then you cannot use that as the bioreceptor. So suitable bioreceptors should be screened first. Sorry. I think this is something that you have to do in any sensor development. The screening of bioreceptors should be done first, whether, I mean, before using the lateral flow, it should be tested in vials, whether it does have a very selective interaction with the antigen. Okay, so so amount of bioreceptor usually higher than the capture bioreceptor. Amount of detection bioreceptor meaning that amount of bioreceptor antibody, for example, on the test line should be usually higher than the capture, which is on the conjugate pad. Okay, should be, that is based on the tutorial paper. So, however, it has to be characterized first. Okay. Um, this one, I think I just skip because if this is actually something, I assume that you know already what is antigen, what is antibody. Sorry. Okay, the difference between polyclonal and monoclonal. So I straight away goes to the nanoparticles. All right, and from the tutorial, you can read about gold nanoparticles, page 17, and also we can use silver nanoparticles, and also fluorescence-based nanoparticles, quantum dots, carbon dots, even though it's quite difficult because for fluorescent, you need to have external reader, meaning that the reader for the fluorescence measurement, because fluorescence are very difficult. We cannot observe you with naked eye. Or if you wish to have non-optical, you can have magnetic nanoparticles. And this one also requires a special reader to check on the test line and control line. So this one is the same one that I have discussed. All right, sample pad and detergent. Okay. So this is commonly used phosphate buffer. And sometimes you need to use detergent to minimize non-specific adsorption. Remember that when you put the samples there, the samples also contain other things, yeah, because it's a raw samples. if you put saliva, if you put serum, they are also contain other proteins that will probably interfere with your uh, conjugated antibody. So you need to have detergent. Sometimes you use surfactant to repel all this and at least help you with the anti-fouling. Yeah. So blocking agent to minimize non-specific agent. You can use also blocking agent. Yeah. For example, BSA. Sometimes we use preservative because we need to keep the lateral flow for 1 year. Okay, so we need to put some preservative, but this right now for us, because we prepare most of the time freshly prepared, so we don't use preservative. And drying sample pad by placing the sample in oven. This is very crucial steps. Okay, as I mentioned, that the lateral flow is very sensitive towards moisture. Okay, so drying and maintain the dry condition is very important. So, okay, conjugate pad, function to preserve dried conjugated nanoparticles conjugated with antibody. In this case, releasing the conjugated nanoparticle upon wetting by sample. So when the sample from the sample pad move to the conjugate pad, and the conjugate pad should be able to uh, move together once they have interacted. Okay, it should be able to move. If there is something wrong with the lateral flow, it doesn't move, then you have to check whether there is some damage or the probably blockage on the lateral flow. So this also requires buffer sometimes. Drying process is very crucial. If it is not completely dry, the conjugated nanoparticles will not be able to move, and you will end a syrup-like solution. So again, the dry condition is mentioned here. So for lateral flow production and maintenance, the dry condition has to be there. So the final product has to be kept usually in aluminum pouch, okay, to make sure that it is dry. Okay, the detection pad is where you have the test line as well as the control line. Facilitate flow, capturing the bioreceptors. It should have non-specific. It shouldn't have non-specific binding. Most important is the capillary flow time, because the targeted time is around not more than 30 minutes. Yeah? So high capillary flow times results in longer interaction, therefore better sensitivity, meaning that um, it shouldn't be too fast, it shouldn't be too slow. Okay, the movement of the conjugated antibody together with the antigen passing through the test line and the control line should not be too long and should not be too slow. Okay, if it is too slow, it might stuck, or if it is too fast, passing through the test line, it might not have enough time to interact with the capture antibody on top of the test line. So how do you immobilize the capture bioreceptor on the test line? Using suitable buffering reagents, stabilizers, alcohol. This one you can ask tomorrow. The capture bioreceptor for the non-competitive has to be 1 milligram per mil. Dispensing process flow rate again. This one is based on if you are using machine. So tomorrow you will learn about manual, how to prepare manually, and we will also expose to you the dispenser machine. absorbance part is the last part, so that one is helps to the capillary movement, as well as it helps to um, avoid the back flow of the solution, okay, of the fluid, sorry. Okay, so this is the whole thing. You have tomorrow laminated card, mounting, drying, sample pad, okay, storage. All right, essay evaluation you will also do tomorrow. So I will quickly give you a few things about um, some new trend of lateral flow. Okay, instead of antibody now, we are moving to aptamer because aptamer is much cheaper. And so this is example of aptamer based lateral flow. Right. Another one is you can use for multiplexing, meaning that on single lateral flow we can have multiple disease detection, depends on different bio marker that you use, okay, bioreceptors. And right, uh, you can combine also, this is the lateral flow, okay, and you can combine with microfluidic before as for the sample preparation or sample pre-treatment before it is introduced to the lateral flow. So it's not just like a normal test strip. You can see that combining with microfluidic, and then you can see it is combined with mask, so you can have the lateral flow as the indicator, and the sample is from your breathe in, breathe out. Okay, so there's a lot of many creative ways of combining lateral flow with existing techniques. Yeah? So it's not just um, very plain test strip. Now we can have a combination of lateral flow with many other um, sample pre-treatment. And the usage of bioreceptors also, it's not just antibody. We can use aptamer. We can also use CRISPR technique instead of just, or we can use DNA. But that one has to be detailed out. Probably the protocol is not similar to the usage of antibody itself. Okay, it has to be designed in terms of uh, control line. What kind of uh, marker that we should put on the control line? It cannot be the same just like when we use antibody. Okay, so I think my time is over. Uh, before I hand over to